I would like to take you today on a journey that I started more than 25 years ago when I had to decide as a junior faculty where to go with my research in environmental health, because I'm actually a faculty in the Occupational and Environmental Health Science Center for the School of Public Health. And so I'm tasked by the state of California with investigating occupational and environmental diseases. And for California, I decided two environmental exposures were of real importance, and they weren't just environmental, but occupational as well. And one was pesticides. So we are actually the state with some of the most agriculturally productive counties. 3 of the 10 most productive are in the Central Valley of California, and we are using a quarter of all pesticides that are used in the whole US. And that's a lot, as you will see. So the Central Valley became my place of research, as well as LA for its air pollution. But today I will only talk about the pesticide work because that is what I started with. And Parkinson's disease was really important to me because as a young medical doctor, I worked in neurology and psychiatry, and I observed somebody with Parkinson's very closely. And I always wondered what caused this disorder. So it had piqued my interest while I was a medical student and a young medical faculty. Oops. Okay, I have to give you my disclosures because first of all, I have, of course, a lot of funding from NIH. Most of what I'm showing here will be NIH funded, but I am an expert in a litigation on Parkinson's and paraquat. So Parkinson's disease, what is it? Well, we know that it is the second most common after Alzheimer's disease in terms of the neurodegenerative diseases, and it affects about 2% of all adults that are above the age of 60. And it is a loss of the dopaminergic neurons in the midbrain right here. And these are the substantia nigra neurons that make dopamine. And when they're gone, they're not there anymore. So you don't see this black melanin substance anymore because these cells are dead. Why do they die? Well, that's the big question. But one reason we know is that they have an aggregation of proteins, and that these are these protein clumps that you can see here that are called Lewy body. And that's how pathologically this disease is actually diagnosed. So eventually we have clinical diagnoses, but eventually you have to actually stain the brain for these kind of inclusion bodies that are called Lewy bodies after the person, the pathologist who first saw them. Interestingly, another pathologist of German origin, Heiko Brack, actually looked at the whole body of Parkinson's patients, not just the brain, and he found these Lewy body inclusions also in the periphery. And he has this theory that possibly Parkinson's actually doesn't start in the brain, or not necessarily, but that it can be actually starting in the periphery, and that these misfolded proteins that then aggregate are transported through the parasympathetic nervous system into the brainstem, into the midbrain, where they affect the dopamine neurons, and then the rest of the brain, where we actually then see all of the non-motor symptoms also arrive that are a feature of Parkinson's. So we usually think of it as a motor disease with tremor, stiffness, inability to move, and eventually falling. But really, the patients very early on may lose their smell, have REM sleep disorders, have a lot of autonomous nervous system disorders, including constipation, depression, and dementia. So it's a very complex disease that's not just in the brain. Interestingly, when we want to study this epidemiologically, we have another problem. And that problem is that these dopamine neurons probably never replenish themselves during our lifetime. So we are basically born with about 500,000 of these neurons. They make dopamine in the midbrain if they're there, right? But then over a lifetime, something may happen and some of these neurons die. So you have hit, hit, hit. It's called the multi-hit theory. And if that doesn't happen, then eventually we have aging. And when aging arrives and these neurons get to be 60 to 70 years old, maybe that is all it needs for them to die. But if there are other hits over the lifetime that diminish their number, we have less and less dopaminergic function. We finally hit this threshold of non-motor symptom dysfunction that starts in the periphery. Nobody says it's Parkinson's yet. We may go on for another 10 to 20 years until we hit this dysfunction that then causes all the motor symptoms of Parkinson's, and we are diagnosing it. So basically, we have an old age disease with a very long prodromal period during which a lot of things may happen so that these neurons keep degenerating. And that is difficult because it's a rare disease. It has a late onset and this long prodromal period. Some people think it's even 30 years long. And so the most common design we are using for the rare disease, of course, is a case control study, because there are not that many large cohorts out there that follow people for 30, 40 years to then see whether they develop Parkinson's or not. And there are no registries in most countries where we can find Parkinson's, which then makes the case control approach also quite difficult. So all that said, what did I do? Well, I wanted to investigate the causes of Parkinson's, and especially environmental causes. So I'm going to concentrate on the pesticides here. But what did we know already about Parkinson's, or what did we learn and I had to also take into consideration? Well, interestingly, there's some literature out there about solvents and metals and head trauma, but most of the literature is actually in this area where we think about protection. And you can see that there's one factor, smoking, that's on the protective side. And there's caffeine, there's uric acid, high uric acid load, anti-inflammatories, physical activity, and certain diets, all linked to protection against Parkinson's without us really knowing, is that true? Or is it because we have this long prodromal phase where patients already have a lot of non-brain related symptoms, and where they may have an upset stomach and they don't drink caffeine anymore, they have REM sleep disorder and don't drink caffeine, or they lose their taste of smell and their addictiveness and they stop smoking. So this is really an interesting paradox in Parkinson's. But I don't want to talk about this today. I want to talk about the pesticide angle. And what about the pesticide angle? Well, pesticides have been linked to Parkinson's in early epidemiologic studies since the 1990s. And already in 2000, there was a meta-analysis, or actually two, published that showed that there's almost a twofold risk increase for pesticide exposure in Parkinson's. What kind of studies did we have? Well, the studies we had were small case control studies. And why do we know this? Well, look at the wide confidence intervals here and the huge odds ratios. And, these were the early studies warning us pesticides may be contributing to this, especially neurotoxic pesticides. But you also see there were a bunch of studies that could not replicate this, and some really large studies, again, that did. Here's a German study that did. So what else do we want to know when we ask the question, do pesticides have anything to do with Parkinson's? And you can see here, of course, we want to know what type of pesticide. We also want to know what type of exposure route, right? Herbicides, insecticides. Insecticides were what we actually thought first off, because insecticides are engineered to be neurotoxic to nervous systems of insects. So they might have a biologic reason to cause neurodegeneration as well over time. But herbicides are another really important subgroup because of paraquat, and I will tell you why that is the case. Here you can see this is from a meta-analysis from Vandermark in 2012, where they showed, yes, a lot of these meta-analytic estimates do say there is something when we are looking at these kind of exposures, even within subgroups of herbicides and insecticides, but nothing specific, right? Herbicides is a huge group. In California, we probably use 50 to 100 different chemicals as herbicides. Insecticides, even worse, there are at least 400, if not more. So are all of them bad? Can we just ask yes, no questions? Well, indeed, we have to go down, I think, to the chemical agent level. And I'm here presenting you a recent review, 2022, on neurotoxicity of pesticides linked to neurodegeneration, where they list at least 3 individual pesticides, rotenone, paraquat, manep, and then two functional groups, organochlorines and pyrethroids. And what I will talk a lot about today is paraquat, and one that they don't even list, which are organophosphates. Pesticides are used more and more. It has started, pesticide production and import has started in the early 40s and 50s in the US, and then steeply, the use steeply increased, and it keeps increasing. Currently, the largest user of pesticides is China, and second largest use is in the US because we are actually a big agricultural nation. And why should we, when we're not farming, be actually worried about pesticides? Well, we should care because according to NHANES, which we know is a US population representative survey, in the year 2000s, at least 96% of the population between 6 and 60 had chlorpyrifos, which is an organophosphate, in their urine samples, and 30% diazinon, which is more used on agriculture, while chlorpyrifos is also used indoors in this time period. Now it's actually outlawed for use indoors, but the chemical companies that are producing it are trying to get it reassessed again and again for indoor use. Well, since the organophosphates have been declining a little bit in the last 20 years in their use, other pesticides have come online. One type is the neonicotinoids, another insecticide, also thought to be quite neurotoxic, and half the US population had some kind of biomarker exposure, according to NHANES in 2015. So we're not out of the weeds if we're not farming. So if we want to know whether these pesticides are actually exposing general populations at chronic levels, therefore maybe providing some of these early hits over a lifetime that affect more and more dopamine neurons until you hit the age when actually Parkinson's is being diagnosed because your motor function displays it as, a diagnosis. What do we need to do? We know there are dietary contaminants that are ubiquitous, and enhanced biomonitoring is supposed to show us whether and how much of these pesticides make it into our diet. There's also home and indoor garden use, and according to this survey in 2000, at least 85% of all US households store at least one pesticide at home. And then there's residential proximity to these applications in agricultural communities. And these communities, even if you don't work as a farmer, your children, your wife, everybody might be exposed. But can we measure it? Can we measure diet continuously? No, probably not. Biomarkers are often one-time events that we can measure them because it's expensive. Can we get any idea of how much we use indoors and on lawns? Well, I show you that we try to do that. And what do we know about these agricultural applications that drift into our homes? We can try to do some personal monitoring. There are wristbands now that actually collect pesticides and other agents on them. But similar to biomarkers, we don't want to wear them every day of our life, right? And we don't want to give a urine sample or blood sample more than once, usually, if we participate in research. So, and these biomarkers need to be either done retrospectively, and we have to have stored samples, or it's quite expensive. So environmental monitoring is actually a way to go if we have information on pesticide applications. And that's what I'm going to show you in a minute. And then I also want to talk about how I did that in California, but I also want to talk about about two tools I used as an epidemiologist. One is gene-environment interactions, and the other is the modern omics tools that our lab colleagues provide with big data analyses that my students are learning. So the study that I conducted and started with NIH funding in the early 2000s is a case control study in 3 rural counties of California, in which we, since then, so it's 22 years ongoing, enrolled almost 850 incident Parkinson's disease patients and about 1,000 population controls, another 200 unaffected siblings and household controls, and we collected biosamples extensively from them and stored them away. We stored blood, saliva, urine, and now also fecal matter. And in the meantime, we asked them a lot of survey questions because occupational handling of pesticides in these counties was widespread. At least 20 to 30% of our population there was farming at one time in their life. So we had these old questionnaire type approaches, right? Have you ever handled insecticides? Which one? On what type of, for what type of purpose? On crops, on plants, on animals? When did you use them? How often did you use them? And what did you use them with, right? All of these questions we asked extensively, relying on recall. And when you rely on recall, you are worried about recall bias or just, people not recalling well. But this is a paper that came out in 2017 in Environment International, where one of my students actually estimated what the effects were on 360 cases and 800 controls at the time that we had collected lifelong occupational information for. And for any pesticide use occupationally, you can see we saw a 2.5-fold risk increase. And then we were able to split it up in these groups, in these application type groups, and even into some chemical ingredients. But organophosphates, I told you, we have hundreds of different types. Actually, 49 were used widely by our participants. And we have only 4.4 cases, 4.4% of the cases exposed to them and 3.8% of the controls, giving us an odds ratio of almost 2, but wide confidence intervals. The other survey we conducted was, from the beginning, was household use of pesticides. Again, a questionnaire. How often did you apply? What did you apply? What did you apply it on? Did you protect yourself, etc.? And then we went through a large, extensive exercise of linking these product names to what was in the California Commercial Pesticide Use Registry, figuring out what it was that was inside it during what years when these pesticides were registered in California for use. And we then listed all these ingredients, like, for example, phenolphthalein, chlorpyrifos, for each single application that was self-reported. And here's the other paper of the student who did all of this work. And you can see that frequent use of organophosphorus pesticides increased risk of Parkinson's about 70%. And when we then adjust for other pesticides or for occupational use, the estimates go down slightly, but they don't go away. And we also have some estimates for the chlorpyrifos diazonons here, individual pesticides, self-reported. But again, you see how small these numbers are. Only 9 cases reported using these and 9 controls. Remember, they didn't report I used the chlorpyrifos. They reported brand names, and we figured out that chlorpyrifos was in them. This is really, I think, what enticed NIH to give us the money to do this, not to do yet another survey where you can, hope that people recall well. This is very unique for California. It's an agricultural pesticide use reporting system that was mandated by law and collects data since 1974 for all agricultural commercial pesticide applications. I got a hold of this database, and my students worked with me over many years to then turn this into a GIS-based tool where we can actually say exactly what in 1 square mile was applied on what type of acreage and what type of agriculture. So when farmers apply either here with tractor trailers or here little planes that fly over and spray, they will report this information to this registry, and we then converted paper maps into land use maps on computers, overlaid this with the information from this POR system, then asked everybody to report since 1974 exactly their addresses they lived at, all of their addresses, and for every single year. And then we plugged that into a geocoder and determined where their home was, and then overlaid that with the information we had from the pesticide use report system. And we could say how many pounds of pesticides were applied around this home within 500 meters or 1,000 meters. And we did this for every single pesticide that was reported to be applied within the vicinity of homes. And that's what we call our POR system derived ambient exposure estimate. We did this at their home address, and this is how we collected addresses. But we didn't stop here. We actually asked them also, where did you work and what was the address of your work? And we did this also at where they worked. And the question, of course, is, does the POR system represent true exposures? And here we got the answer from another study done by EPA, actually, in California, where you have this little town, Parlier, and these are elementary schools. And on top of the elementary school roof, they put monitoring equipment, air monitoring equipment, and collected samples that they analyzed for almost 50 pesticides, and then for a whole year. And then they compared what they measured on top of these schools to what was applied according to that pesticide use report system within about an 8-mile radius of this little town. And this is what they showed. So this is the weekly sum of diazinon applied, and this is what they detected in their air monitoring. The bars are what they detected in their air monitoring system. And you can see that there is a quite nice overlap between the monitoring system, the bars, and what was reported in the pesticide use reporting system, right? When there's agricultural applications, you can measure it in the air. That doesn't mean that people breathe the air, but we are at least closer to saying, yes, we have some representation of exposure here. So now I'm coming to the meat of this with the gene-environment interactions that we used to establish whether the risk was increased. But in order to do gene environment, you need to have a gene, and you have to have also some exposure. And the question is what exposure to choose. And first we chose paraquat because, first of all, there are animal models of paraquat that make animals look like they have Parkinson's disease. And that's possibly because paraquat looks similar to the one known toxin, MPP+, that's a derivative of MPPP, a contaminant in synthetic heroin that wipes out your dopamine neurons if you give that to primates or to humans. And you can see here in this odds ratio, that is one of the first papers we wrote on pesticides. Paraquat and Marneb together increased the risk in our case control study by about 80%. So how did we use this for looking at gene-environment interactions? Well, there was one study from Washington where Dr. Checkaway and his colleagues looked at the dopamine receptor, dopamine transporters This is dopamine in the synapse, the neuro agent that needs to be there in order for our dopaminergic system to work. And these are the synapses of these dopamine cells that actually die. And this dopamine gets released into the synapse and taken back up by the dopamine transporter. And interestingly, this dopamine transporter is supposed to also take up toxins, including paraquat. So the idea was that if you have a highly functioning dopamine transporter, then you might take up more of the toxin from the synaptic cleft as well. And this was the result that was presented by Dr. Chekaway's group. You had to have a high functioning dopamine transporter, and you had to be exposed highly to pesticides in order to have an almost 6-fold risk increase in Parkinson's. And it was less if you had only one allele of that functional variant. So I didn't believe him. He called me and said, Beate, you have to look at this again. You know, we are seeing these incredible interactions. And it took me another 3 years to finally say, okay, yeah, I will look into this. We have enough data now to do some gene environment interaction study. And this is what we saw. And remember, what he saw was a 1.6 and a 5.6. And this comes pretty close, only in the high exposed group, and only if you have the functional variant of that dopamine transporter is your risk actually increased. Under no exposure, that transporter does not put you at increased risk at all. So you need the exposure and you need the genetic component that tells you something about protein function, in this case, the dopamine transporter, to find that, yes, maybe there's a risk increase in Parkinson's. I think this is probably the first ever gene environment interaction in pesticides that was actually replicated. So we wanted to do more than that and not just stop with paraquat. So we looked at other pesticides. This is a paper that came out in 2014, also by one of my students. And he just said, I'll look at every single organophosphate. And this was his list. We used the ambient exposures and we used the workplace ambient exposures and both together, right? The addresses of residences and workplaces. And you can see almost all of his estimates are increased. Not all confidence intervals exclude the one. He has slightly higher risk in workplace address exposures and even higher ones when the two came together. But what does that tell us? Well, when we are then also looking at mitochondrial disrupting organophosphates, and mitochondria seem to be one pathway of mitochondrial dysfunction, seems to be one pathway that is implicated in Parkinson's, you can see that we are starting to see a little bit of a dose response if we're just doing pesticide counting. So if you're exposed under 7 or up to 7 organophosphate pesticides around your homes, your risk is maybe 2-fold. If you're exposed to even more, you're going up to a 3.4-fold risk increase. Okay, that is not recall bias. This is really based on where you told us you lived and your exposure since 1974 and prior to Parkinson's, right? But we can now use our gene environment interaction tool to do something really cool because we know something else about organophosphate insecticides. And what we know about them is that they first get turned into an oxone. Sorry about this. They get turned by the P450 system into a toxic metabolite, an oxone, which is really neurotoxic. And then by an enzyme called paraoxonase 1, they get detoxified and excreted. We also know that in human populations, you have poor metabolizers and ultra-rapid metabolizers and everybody in between. And there's about a 40-fold difference in how quick your enzyme is metabolizing organophosphates. And there is a group of people, about 7% of the population, who are unlucky enough to be in the poor metabolizer category. And you can actually capture that with these PON1 genotypes 55 mm, and 192 QQ. And these are assays showing how slow your enzyme is. This is the slowest, right, compared to all the others. We use this genetic information to inform our next set of analyses. And the first set of analyses we did was use our most prevalent use of organophosphates, diazinon, chlorpyrifos, parathion, and then use what we know about metabolism capacity based on the genetics. And you can see that in every single instant, our PD risk goes up when you are exposed and you have a functionally low functioning detoxifier. So this is low functioning, medium functioning, high functioning detoxification. And you can see it here for chlorpyrifos, the risk is threefold when you're low functioning. It's about 1.6 when you're medium, and there's very little risk when you have a fast enzyme. So I think this is really convincing that we're not just looking at some artifact. And then I told you that we also had our survey of OP household pesticides, and that my student went through all of these pesticide names in order to figure out which ones were actually having an OP in them. And chlorpyrifos was widely used in these products in the 80s and 90s for indoor use against insects. And you can see here that if we have people reporting never use or rare use of pesticides versus frequent use of OP pesticides, not any, but OP pesticides, it is really the slow metabolizers that have the increased risk, 2.6-fold increased risk, confidence interval excluding the one. So we have two completely different sets of specific pesticide exposure assessment tools that we applied. One, what was sprayed by agriculture around your home? The other, what did you use in your home yourself? In both cases, gene environment helped us to show that, yes, you had to also have some susceptibility by not being able to detoxify these pesticides when you were exposed. Interesting, I find this one of our most compelling other arguments that what we're seeing is really relevant. We actually then had the chance to do some methylation analyses in Parkinson's, and we found, we did an EWAS and found these differentially methylated CpG sites. And in what way differentially methylated? Well, what we did is we looked at these epigenetic changes in people who were OP pesticide exposed, and we had this whole measure of 1 exposure, 2 exposures, up to 50 exposures as a continuous variable over their lifetime. And we found that the overrepresentation in gene sets that were differentially methylated in blood, not in the brain. We don't have brain cells, right? But these white blood cells showed nicotinic acetylcholine receptor signaling pathways and muscarinic acetylcholine receptor signaling pathways as the main differentially methylated pathways. Why am I so excited over this? What do OP pesticides do? They inhibit the acetylcholine esterase. So these are exactly the pathways, and that's what they're engineered to do. These are exactly the pathways that then our cells are reacting against when we see too much acetylcholine, because the esterase is inhibited and acetylcholine, that neurotransmitter, builds up in our blood, builds up in our brain. And we can see the signal of long-term exposure. Our white blood cell genetic has actually compensated for this long-term exposure. So we also are using now metabolomics tools, and this is another paper I wrote with a really talented graduate student of mine, Chi Yan. And what we did here is we used the same GIS pesticide exposure tool, and we had serum samples. We did an untargeted metabolomics screen with Emory, with the Emory group of Dr. Dean Jones. and we analyzed it according to 3 types of pesticides, the organophosphates, pyrethroids, and organochlorines, and then saw what in the metabolome of these, and these are only control subjects, by the way. Nobody has Parkinson's. We're just using our controls. And then looked up what the metabolites represented that was different in the long-term exposed versus the other individuals. And these are the pathways we picked out from the metabolome: inflammation related to Parkinson's, oxidative stress, very important for brain cells, and mitochondrial dysfunction, which for Parkinson's has been claimed again and again to really make a lot of sense in terms of pathogenicity. The mitochondrial function is impaired. Remember, this is not in Parkinson's patients. This is what pesticides do in healthy controls or neurologically healthy controls, right? So they impact these pathways that we also find to be impacted in Parkinson's patients from other research, from genetic research, for example, or from models. So here we are, we have mitochondrial dysfunction, we have oxidative stress, we have these pesticides again, and we picked it out in our screen, in our metabolomic screen. What are we doing now? Well, I'm going to show you a few more slides before I like to get your questions. We are now doing a PWAS, a pesticide-wide association study, and these are all of the pesticides that a young, talented colleague of mine, Dr. Kim Paul, has picked out using this kind of untargeted screen of all chemicals that have been applied in the Central Valley, except for the ones that we already had investigated. And she can now use an overrepresentation analysis to pick out which ones are the ones that we're seeing related to Parkinson's, the drift-prone ones, the highly hazardous ones, the acute toxicity ones, the choline esterase inhibitors, that's our organophosphates, and what already has been described by the literature as bad actors. And here they are again. So from epidemiology, we cannot say much more than, okay, we picked them out. We have our PEG study, we picked them out there. What I wanted to show you is that we now partner with a group of Dr. Khurana at Harvard to look into human pluripotent stem cells, that we apply these pesticides that we picked out, the 70 pesticides we picked out, we apply them now to in this pluripotent stem cell model to genetically engineered human stem cells to see what it does to these human stem cells, these different agents, and maybe pick out the different bad actors. And this is a paper that's about to come out. And all I wanted to show you, there's a lot going on with making these stem cells and then testing them. But what I wanted to show you is we have negative controls, we have positive controls, and out of the many that he tested, these are the ones that in the stem cells show toxic effects. And thank you. And I'm almost done. So what else are we doing with Dr. Paul? We just published this paper in Environment International, where we linked these same 70 targeted pesticides to other tools that are out there. For example, the Tox21 Toxicology in the 21st Century program gives you lots of pesticide-induced activity assays, and we're exploring these now. And we are also going to another big data set database, the toxicogenomics database, a comparative toxicogenomics database, where we can identify networks of genes that are actually linked to pesticides in the literature, and also can do relevant gene ontology annotations. And this is one of the images that you'll find in this paper, where we can see which are actually the genes that we can pick out that are most commonly interacting with the pesticides in the literature. And this is not epi-literature. There's a little bit of epi-literature. It's mostly cell literature and animal, genetically engineered animal literature. And then we can, generate these nice plots to visualize what we've been finding. And we can also see how different assay responses, such as response to oxidative stress, regulation of neuronal deaths, in these assays actually is found to relate to these pesticides that we have picked out. Here, chlorpyrifos that I showed you throughout, very strongly related to some of these mechanisms that you can pick up in these different tox databases. And you can then visualize this in this manner and see, you know, how many different assays are actually related to this kind of, and forming a network in this case. And this is basically my last slide on research. This is what we're doing now. We actually go to the next step in terms of Parkinson's, I believe, the next step. And that is to see what happens in the gut and ends up in the brain, whether or not that's related to neurotoxicity that we end up with when, you know, neurodegeneration and neurotoxicity that leads to neurodegeneration, and whether, for example, the microbiome and the immune system are participating in this action. The microbiome, because we know that there is gastrointestinal problems in Parkinson's patients up to 30 years prior to the motor symptoms, but also because we found that alpha-synuclein, that aggregated protein, is found years before it's found in the brain, in the gut. It can be through the vagal nervous system, the parasympathetic nervous system of the vagus, transported to the brain, and then possibly built a foci around which then other alpha-synucleins also aggregate and build up Lewy bodies that eventually kill these neurons. And so that's what we are investigating now, these links to the microbiome. And we're not only looking at alpha-synuclein and the microbiome, we are also looking at the microbiome as a big detoxifying organ that we have, meaning pesticides can actually be metabolized, of course, by the microbiome, and they can be metabolized to a more toxic or a less toxic agent, and that probably depends on your microbiome composition. All right, and that's what we're doing now. And I think that we need a really strong scientific story to set standards and to have public policy go forward on pesticides. They will not disappear from the market, but hopefully we can identify the bad actors and get some regulation on them. What we eventually, I think, need to lobby for is to regulate pesticides like pharmaceuticals and make pesticide companies monitor humans and the environment and do something that was coined by these authors, pesticidovigilance, after pharmacovigilance. I really like that word. And I wanted to close this with one of your own, E.O. Wilson, who said, if insects were to vanish, the environment would collapse into chaos. Insects support terrestrial food chains, serve the planet, chief pollinators, and crucial decomposers. And yeah, I think he had quite a point there, you know, just sterilizing our environment will not get us there. And these are all the really talented people I work with, and none of this would have happened without my colleagues in neurology, my colleagues in epidemiology, and all of my students. This is a very early picture, 20 years ago, of the first students who worked with me. And none of this would have been done with all of the funding and all of our collaborators. Thank you very much.